simply doesn't exist for them. Okay, they just don't have it. So, we know that the term disease was actually coined by Pasteur, which seems kind of weird. I'm sure people said sick before, but the word disease is a word that Pasteur put together. And that was just to describe, you know, the spoilage in beer and wine, because remember, that's how we started this whole course, right? A virus is a disease-causing agent. That is one way to describe them. And so they were discovered a while back, but this is well before more powerful microscopy. They are in the nanometer range. Do you want to start by actually looking at the coffee bean thing one more time? Does that help you?
Yes.
Okay, let's do that. Sorry for the break here. Let's do that. Amoeba proteus, first of the cells that they used to show us human eggs, skin cell with a micrometer range. Look at that. And then, sorry?
Can you see human egg with your eye?
No.
No, it's too small.
Yeah, you can. Okay, so skin cell, red blood cell. Finally over here, Baker's yeast. Okay, so this is a microbe. Can't see yeast. And now you're starting to see E. coli. Okay, so this is in the micrometer range, as is mitochondria, as are lysosomes. Okay. All micrometer. Now we're into virus, and this is nanometer. And so, see that phage right there? Remember we talked about transduction? Okay, that would actually be a virus that infects bacteria. It would land and squirt its DNA in and all of that. Look how much smaller it is. Okay, nanometer range. As we keep going down, and you know, there's different size viruses, but they're all nano. As we keep going down, let's get here to rhinovirus. This is the causative agent of the common cold. And you notice it's the same size as a ribosome. We don't have that. If you contrast eukaryotic cells and bacteria, we know that bacteria lack nuclei, right? They're too small to have a nucleus. They're too small to have mitochondria. They're the same size as mitochondria. They don't have rough endoplasmic reticulum. They don't have smooth endoplasmic reticulum. They don't have lysosomes. But they do have ribosomes. All cells do. A ribosome isn't an organelle, right? It's just 2/3 RNA, 1/3 protein. A virus doesn't even have that. They're too small for that. Okay, so they're a stripped down version is what we'll see. But having said that, you can see this green, that's the antibody, right? That we will end up making. So this will be our, you know, final leg will be our immune system. And we will create antibodies against different pathogens, whether they are viral or bacterial. Keep your eye on that green thing. Okay? So you can make antibodies to these little viruses. See that? And you see how much smaller the antibody is? It's just a little speck, right? And that's on the virus. So when we go this way, and you're saying that I made an antibody to bacteria, and it gets coated with antibody, yes, it does. Okay, yes it does. And so, you know, if you have a yeast infection and you have antibodies against it, that's a huge response you can mount to that. Do you see that? Okay, so we want to think about this and look at it. It really helps, I think, to keep looking at this every once in a while so that you know where you are. Okay, it really is kind of a map for us. What are we talking about today? Are we far away? Are we really tiny things? What's going on? So we'll start this conversation with saying that viruses are really, really little. They're in the nanometer range. So now when I tell you that here's Pasteur studying rabies, which is a viral infection, and Mayer, who was studying tobacco mosaic virus. Now we're not interested in that in healthcare, but he was one of the first pioneers in studying virus. And so tobacco was a huge crop back in the day. This is 1880s. And so it would develop these weird spots that start like this, pretty soon takes over the whole leaf and destroys the plant, and so that whole crop would be useless. So he was tasked with, can you figure out what this is? Rabies is a fatal viral infection because it affects the central nervous system and it leads to an encephalite, so an inflammation of the brain. It is fatal. We're not used to thinking about that because all domestic animals have been vaccinated against rabies. Okay, but if you get bitten by a wild animal, you have to call the doctor right away. Okay, and if you can, they tell you bring in the animal, right? They want to know this thing doesn't have rabies because it is in fact fatal. So he was studying people and animals that had rabies, Pasteur was, and the two of them independently tried everything they knew about culturing bacteria on these two systems, on what they saw in the tobacco leaves and what was seen in the diseased grain. And they were unable to isolate it. Right? So all the things we know to do for bacteria, they couldn't do it. Pasteur also took the brain and dried it up, lyophilized it, broke it up, then reconstituted it a little bit and put it through a filter. And they already had filters that were in the micrometer range that would stop bacteria. Okay, remember we said that filtration was a way to sterilize, remember that? Okay, but the thing about filtration is that we always have to look at what's the pore size. If you can stop micrometer size things, then you most certainly will stop bacteria. But if I want to stop virus, it has to be nanometer. Okay, that's 1,000 times smaller. That's a big ask. So what would happen is that you would pour the grain that was smashed up through the filter, and the stuff that dripped through was still infectious, meaning he could take this and put it into an animal and they would develop rabies. It was like, huh? And both of them realized that they couldn't stop whatever this was with ethanol. So we just talked about this in first lab, and second lab you will too. We already put the organisms on the phenylethanol plates, and we said that that would kill off the gram negatives, and you'll see it does a pretty good job at that. Okay? There is a concentration of ethanol that will kill gram positives too. Okay? So it just has to be high. Okay? They have a built-in block, that very thick wall, right? Protects their plasma membrane. But they couldn't stop this with ethanol. And they couldn't culture it in media that supports bacterial growth. And I have an asterisk here because we know that that's not true even for all bacteria. Remember that things like Treponema pallidum, the causative agent of syphilis, it won't grow in anything other than human gonads. Right? So we can't figure out what it wants. Okay. But most bacteria, you can figure out what they want and add it to the media and they'll grow. Okay, so they couldn't kill it with ethanol, they couldn't grow it, and the only way they could get more of it was to grow it in a cell or tissue. So here I show you some eggs. You know that eggs are a cell. And for Mayer, he could grow it on the surface of another plant. So then they just said, I don't know what this is. This is not bacteria. I always ask this question. Right? And it probably will come in the form of multiple choice. How did they know this wasn't bacteria? Right? They couldn't see it. They couldn't stop it with ethanol. They couldn't put it through the filter. They could only grow it in other tissue. They couldn't culture it. Like all the things that would be correct for bacteria are not working here. So they knew, we don't know what it is. It's not bacteria. That was their conclusion. And it took a long time to actually see them. It requires electron microscopy. In this PowerPoint, there will be plenty of pictures of virus. Every last one of them, that is a real picture, will have an EM at the bottom of the picture, indicating this is an electron microscope. This is not light microscopy that we use in lab. And they're much, much smaller. So it was Pasteur who came up with this term virus, which apparently means poisonous substance in Latin. I don't speak Latin, so I just take their word for it. Virus. But now that's what this means. Virus, okay, a poisonous substance. because it's describing a disease-causing substance that is not bacteria. All viruses are pathogens, all of them. Some are mild, some are more serious, but they are every one of them pathogenic. And they are fundamentally different from cellular forms of life. So here what you see in this picture, Mastodina virus, this is another way to get a common cold. This is phage, bacteriophage. This is the surface of bacteria here. Okay, do you see how much smaller they are? Okay, and we'll see that this is where they inject their DNA. Right, right there. This is a very important slide in the context of virus. And so I will have this on the exam, either exactly like this with the rows mixed up, or it could just be one of those, the following is true of bacteria, virus, both, neither, okay, etc. You've seen those kind of questions from me before. So it just takes going over this. At the moment, it's going to seem like you have to memorize it, but when we get through the PowerPoint, it's just part and parcel of what you will have to know. Okay? So there's 3 columns here. The first one is typical bacteria, and that's the only one that you need to know. Okay? And then virus, and we need to know that as well. But in the middle are very strange bacteria. Ricketsias and chlamydias. Ricketsias and chlamydias, the thing is they're very small, and they don't behave the same way as normal bacteria. I don't want you getting tripped up on this column. Okay, we want to know the first column, typical bacteria and virus. I will, however, on every row, talk about the chlamydia and the rickettsia, because I don't know what the future holds for you if you will ever have a course that mentions them again. However, we got to know this, chlamydia, it's a rising STD. Your patients with potential STDs, I bet you it's chlamydia. It's number 1. So we may as well know a little bit about it, okay? But I'm telling you that for the exam, don't kill yourself knowing the stuff in the center column, okay? Because I am not going to task you with it, okay? All right. So starting from the top, intracellular parasite. So has to be inside of a cell in order to live. See, let me back up just a minute here. It's almost impossible to describe virus without using words that mean alive. And soon we'll see that they're in this nebulous region where they're not quite like living things, but they're obviously dead used to be living, and then the only other word I have, the only other term I have is non-living, which is not that satisfying, right? Yes.
Also, that brings the question, how do you kill a non-living thing?
Well, you will know in moments. Okay, so it's a lot easier than you might guess. Okay, do you remember our resistant, most and least resistant? Who was at the bottom? Virus, both kinds. Okay, because they have no way of keeping themselves, like homeostasis, this fight you're fighting right now, keep your body temperature against this, they can't do it. They got nothing. Except the will to infect.
They're just like a capsule of RNA, basically.
Oh, we are getting there. You're champing it.
I have a little question.
Yes.
So, since it has RNA, it has.
They don't all.
They don't all have RNA.
Nope.
Okay, I'm gonna start
All right. All right. So here's the thing. Intracellular parasite. Do you have to be inside of a cell in order to exist? For most bacteria, no, they don't need that. Okay? For all virus, the answer is yes. Every single one of them must be inside of a cell in order to exist. The ricketsias and chlamydias, this was the little issue. It took them a while to figure them out. In fact, they figured them out after they figured out virus. Okay, because they're weird bacteria that need to live inside of another cell. Okay? How about if you have a plasma membrane? Now you know that there's 4 basic structures of any given cell, whether you're bacteria or eukaryotic cell like yours. plasma membrane, number one. Okay, the boundary to the cytoplasm, all have it, and it has specific functions, right? Okay, and I want us to remember something, that a plasma membrane is far more complicated than just a phospholipid bilayer. Okay, a phospholipid bilayer certainly is the beginning of it, but remember of all the proteins that are embedded, that those are the ones that are actually carrying out the functions, right, of the plasma membrane. Okay, so typical bacteria, even these weird ones, they all have a plasma membrane. No, over here for virus, they don't have a plasma membrane. We just said that's like one of the things that every single cell has, they don't have it. Okay, do you divide by binary fission? Now, you'll notice that this says bacteria and virus. Those are our two categories. If this was about a eukaryotic cell, you'd have to say no as well, right? Okay, but we know that bacteria, that's what we define as growth, is binary fission, and normal bacteria and Rickettsia and chlamydia, yes, that's how they divide, is through binary fission. That is not what goes on for virus. They do not divide that way. Okay, can you get through the filters? Okay, so filters that would normally stop bacteria, they're in the micrometer range. Yes, you can stop bacteria, so they can't get through that kind of filters. Neither can Rickettsia because they're kind of big, but the chlamydias are smaller and they can slip through those filters. So that's a little tricky. All virus can get through bacterial filters. Okay, that's something to think about. That's a problem. Okay. How about, do you have both DNA and RNA? Now, any given cell, we know that the genetic information is stored always in the form of double-stranded DNA. It might be many single linear strands like we do, what we have. It could be one circular, like bacteria have, okay, always. But the DNA really doesn't do anything, right? It's always just sitting there. In order to express the DNA, you use messenger RNA, tRNA, and ribosomes, which are 2/3 RNA, right? So any given cell has DNA and RNA. Are we in agreement? Okay, so that is absolutely true of these kinds of bacteria, all of them, okay? Not true for virus. A virus is either a DNA virus or it is an RNA virus. It is not both. It is never both. One or the other. That's it. Okay. Can you make your own ATP? Okay, and we just went through this. So, can you take some sort of food, glucose, whatever it might be, and you break it down, pull apart those carbons, okay, take the electrons, and somehow create ATP, either substrate level or oxidative phosphorylation. Can you do that? All bacteria can do it, one way or another. All 5 tubes have some mechanism of doing that. So do the Rickettsia, chlamydia, no. Kind of freaky, okay, but none of the virus can make their own ATP, none of them, not one. Do you have ribosomes? Any given cell has ribosomes. Yes, even the weirdos, not virus. Why not? Why not virus? Too small. They're too small. All right, so, you might just be thinking, I can't wait to get out of this class, and I'm not even sure I want to go into healthcare. Okay, good. I get that. I hope I didn't do that to you. But I hope you remember this. Sensitive to antibiotics. In fact, to take exam 3, I will ask you about this question. So, can I attack typical bacteria with an antibiotic? Absolutely. Yes, okay, I've got 5 different classes. Within each class, there might be multiple targets. Okay, I can kill them with an antibiotic. What about resistance? They're not resistant to everything. And I can do a drug cocktail and slam them against the wall, right? Let's go ahead, build it all. Efflux pump. They can't. They can't do that antibiotic, right? We get that. All right, so, and even these guys, we'll just figure it out what you have, and we'll get you the right antibiotic. You can't use that here. You can't use that here. And so I'm going to pause here for just a second because 60% of infectious disorders in the United States are viral. 60%. So, and you know very well you've been given an antibiotic for that thing, and it was viral. You're not doing anything except training your normal microflora to be resistant. You are killing off the sensitive and you're leaving yourself the resistant behind. That is what you're doing when you're taking an antibiotic during a viral infection. I will 100% ask this question. I will ask, why can't you use an antibiotic on a viral infection? And I expect to hear all 5 categories, all 5 classes. You give me 1, that's 1 point. You remember 2, that's 2 points. Okay? You need to know them all. You're going into healthcare, this is one of your tools. You've got to know all 5 classes of antibiotics. That's just one of those things you're bringing with you. Okay? All right. You can't use that here. At the bottom, though, this is something that we will come back to when we go into immunology. Interferon is a secreted protein. And you know how that happens. Right? A secreted protein. Cells that are infected with virus will secrete interferon to warn the neighboring cells so that they can help themselves against viral infection. Okay? That works on virus, yes, over there, but that does not work on bacteria. You know why? Because it wasn't their plan to get inside anyway. Okay? Bacteria do not need to be inside of your cells. A virus needs that. They don't have a metabolism. They need to be inside of a living cell in order to propagate. Remember my example I've given you many times, 2 slabs of meat, bam, squirt some bacteria on one, smear it, come back later, that's gonna be a mess. They will have broken that down. All 4 carbon compounds are gonna have a slurry, right? Over here, put the worst virus you can think of. A slab of meat is dead. Dead things don't have a metabolism. Dead things don't make ATP. That's it. That virus cannot grow. It can't, there will be no more than before. In fact, it will just begin to decay. I feel like there's a pending question with you. No? All right. Yes.
What do you exactly mean by sensitive to interferons?
I mean that they will be destroyed by it.
Okay.
Okay, why don't I just say
How
that?
does interferon destroy a virus?
I'm sorry?
How does it destroy a virus?
Interferon?
Yeah.
So when we get to immunology, we're going to spend some time on that. But what happens is that the virus needs to take over the cell's machinery and it won't be able to if interferon has been secreted.
Does it like sort of like a competitive inhibitor?
No, it's not that. No, it triggers in the cell next door a bunch of enzymes. Yes?
How is the virus able to actually differentiate between being a non-living cell and a living cell?
It isn't.
Wow.
Yeah, it isn't.
It just can't do its
It just
thing
can't,
to
it just doesn't have the dead cell just doesn't have the stuff that it needs to actually get into it.
Yeah, yeah.
We'll get there, we'll get there.
All right, so the first virus ever seen was the tobacco mosaic virus, and you see this is an RNA virus, and it's wrapped up in protein. You see why I'm always talking about proteins? Because they're everywhere, and it's always the thing. So here's the RNA, and it's wrapped up in proteins. You can see there's a bunch of subunits, but together it makes this helical capsid. It is a protein coat. The purpose of the capsid is to protect the nucleic acid. Okay, this one happens to be helical. And this is an actual electron micrograph of it, so it's this long rod. Happens to look that way. Rabies is different. This is a bullet-shaped. Okay, this is a bullet-shaped virus. It also is RNA. It has an RNA genome. It has on its surface, on top of the RNA, a bunch of protective capsids, a lot of proteins protecting the RNA. But it also has a phospholipid bilayer on the outside here. This phospholipid bilayer it stole from its last host. It is not a full plasma membrane, it's just a little pinched off piece of lipid. when a virus like this is an enveloped virus, when it steals a little pinched off piece of plasma membrane that belonged to a cell, well, the immune system is trained before these cells are released out into the blood, into the tissues, they go undergo some training. And they cannot respond to self or they get deleted. So this is a nice little way for them to kind of hide from surveillance. Okay, they're carrying with them plasma membrane from the last cell. So here's just a comparison of size, but it's inferior to the purple chocolate, or sorry, coffee. So here's just size images, all right? So the next several slides are about the general characteristics of virus. Virus differ very, very much, but we will talk about the generalities of it. And I usually have a page that kind of sort of gives the bullet points for you to flesh out. Okay? I think I already have that on the virus worksheet. Okay, I can get started on that. So they're all of them. Every single kind of virus is genetic material in a protein code. Okay? Why can't I just say DNA? Because some are DNA and some are RNA. So I have to be really like generic. Some sort of nucleic acid, okay, some sort of genetic material wrapped in a protein coat called a capsid. If that's all they have, so this one here, the nucleic acid wrapped in a capsid, nucleic acid wrapped in a capsid, this is helical, so it'll be all around it. It's just showing you kind of a cutaway. All right, then that is called naked or non-enveloped. But some have a nucleic acid, capsid, and then the phospholipid bilayer they stole from the last host, whether it's helical or this shape is called icosahedral. It's pretty cool. It's 20 different triangle faces to form that shape. Very complex. This has a phospholipid bilayer on the outside, as does this one that's known as enveloped. A naked virus versus enveloped virus. And that's true for any kind of virus. They fall into one or the other category. Whether they are naked or they are enveloped, they have on their surface a protein known as a spike. So we see it here on this naked one, we see it here on the enveloped. That spike is really specific in shape. It's a protein, so it has a shape, and it will dock on a particular receptor. Okay. Receptors belong to cells. That capsid, okay, can be enveloped, as I said, or naked, but the shapes are polyhedral or icosahedral, like this one, a 20 base triangle thing. Helical, as we talked about, bullet shaped, as we saw with rabies, and also complex. So here's an icosahedral that's naked, icosahedral that's enveloped. That's herpes. We have helical naked, helical enveloped. Like this is Ebola. This is the flu, influenza. Conical, like this, include not only rabies but HIV. So these are different shaped capsids, but they all do the exact same thing. Complex simply means they defy categorization because it's like, what do you want to say about this? It has an icosahedral head, a helical shaft, and then tail fibers. It's just too complex. Okay, and then the smallpox is more like a brick. So there's this one for complex. Yes?
Can the same virus be naked and enveloped?
No.
So like have two different versions? No. So is there a specific thing that allows it to get or gain that lipid bilayer? Is there
Yes.
some enzyme that it has? Or?
Yep. So the genetic material is either DNA or RNA. It is never both. However, amongst the RNAs, we have one strand single-stranded RNA. We have 2 strands. They insist on writing DS as though it means double-stranded RNA, and I hope you join me in like heckling that, like what as if, because there's no way, you know that can't happen. Okay, there's a 2'-hydroxyl on all nucleotides. I know, I did, it's really good. So 2 strands of RNA. Then we can have 2 strands of RNA with associated proteins, and you'll remember this when we get to HIV. With DNAs, they can be single strands, double strands, a single circular strand, double-stranded circular, 2 strands with the ends covalently sealed, DNA with terminal proteins. I want you to memorize this. No, I don't. No, it's either RNA or it's DNA. That's it. Okay, never both, and not even in the same format. Hello? Just because you're RNA, I still don't know what you are. I don't know what you are. Are you single? Are you 2? Do you have stuff associated? I have no idea. I need more specifics. If you're talking about any given cell, whether it's yeast, right, your DNA is a double-stranded helix. That's it. All cells. Bacteria, double-stranded helix. It is a circle, but it's a double-stranded helix. Yes, Amelia.
If it's a double RNA with associated proteins, are both RNA strands the same?
Yes.
Okay.
Well, no, I mean, they're complementary.
Okay. So the okay, but they can't go together because they're
Right. All right, so let's look at this. This is HIV right here. So this is its capsid. And I want us to look at it for a moment because you see that it looks like chicken wire. That's not conical or bullet shaped. You see how it's really more like a chicken wire thing? Okay, but if you look at any given hexagon within that chicken wire, hexagon has 6 sides. Each side is composed of 2 alpha helical proteins, so that means 12 proteins all the way around, right? Okay, the site of protein synthesis is what? Right. I sure did somewhere. Ribosomes. Because, bacteria make protein all the time. They don't have roughly. Okay. So the site of protein synthesis is ribosomes. Do viruses have ribosomes?
No.
Okay. And is protein synthesis, is that anabolic or catabolic? Anabolic. Costs you energy, doesn't it? Do they know how to make their own energy?
No.
Okay. So you're asking the cell to make you 12 proteins for each hexagon, and I haven't the faintest idea how many hexagons are here. You're just saying, I'm going to take all this stuff. And because they don't have a metabolism, they didn't break down their own protein into little amino acids from which to begin. They're using the cell stuff. Okay? Here's the thing. Viral infections cause fatigue. Okay? That's how you're like, okay, I'm run down. Okay? I'm just gonna take a wild guess. Okay, 60% of the time I'm gonna be right. Alright? Unless it's, the real issue that's killing us, which is what? I'll keep reminding us. Heart disease. Okay, yes. Given that you don't have those 2 things and you feel run down, could be virus. That's what happens. Okay. So they can be super complicated. And yes, I just showed you the bullet shape. The icosahedral is just as complicated. Notice that this is made up of 4 different proteins to make one of these triangles, and there's 20 of them. That's a lot. The helical stuff seems like, oh, well, that's no big deal. This is all the little subunits just to go around once. And this is multiple rounds, right? So, in other words, they're all costly in terms of ATP and use of building blocks and all of that stuff. They didn't bring anything with them. Yes.
Sorry.
No, it's no problem.
So, on DNA and RNA, they both have a sugar backbone, correct? Like that sugar phosphate backbone. So, wouldn't that mean that anything that would dissolve sugars and stuff would be like a pretty good agent versus virus?
Well, yes and no. Okay, so what does the virus want to do? It wants to get inside your cell. And so I'm just going to apply something that would destroy nucleotides. Well, I got them too, right? So like, got a little problem here. These are the issues. Like, you gotta recognize, like, I'm built of the same stuff. I have proteins, I have nucleotides, and what we know certainly about cleaning agents, and even antibiotics, they don't know what they're doing. Okay, right? They just go and do the thing. And so, that it just gets tricky. Okay? Alright. So, naked or enveloped spikes on the surface, okay? They are all of them, so we have, all we've done so far is genetic material wrapped in protein coat. Okay, those protein coats, capsids, can be different shapes, icosahedral, helical, conical or bullet, and complex. Okay, which just by categorization takes a lot of energy to do. Okay, point number 2. They're all obligate parasites. A parasite is a situation where an organism and or whatever this is, virus, is on the take at the expense of the host. Okay? They've got to be inside of a host cell in order to reproduce. Sometimes I say replicate, because reproduce is kind of a living word. Make more copies. Okay? It's really hard not to slip and keep using words that mean living, right? Okay. They land with their spike on a specific receptor. They get brought into the cell. Okay, we've gone through endocytosis before. We talked about eukaryotes and how they bring material in. Okay, so they come on in. We'll talk about this. The lysosome breaks apart the capsid, releasing the nucleic acid. In this case, it looks like it's RNA. Okay, gets translated. Okay, you know, if my small ribosomal subunit sees an AUG, it comes a running. Right? And then we get started, tRNA, large subunit, let's go. And suddenly you're creating all this stuff, and it's like, wait, what is that? Okay? Now, look at this thing. It was a enveloped virus. You see this? Okay. Some of these proteins are going to be on the plasma membrane, and those are made in a specific way. Rough ER, Golgi, vesicles going over here. We've told this story before. Meanwhile, it's RNAs being copied, okay? And so is the capsid being made, right? And I'll describe this part here. And then, look at this. It pinches off, carrying with it a plasma membrane that belongs to a cell that was yours, a cell that your immune system is designed not to destroy. That's tricky. Okay? So, why do they have to be inside of a host cell to reproduce? Because they lack all metabolic machinery. All they are is genetic material wrapped in a coat. Okay, so all they need to leave is like, just make me a bunch of more genetic material and make me more coat. That's a protein. They don't have ribosomes. Okay? They don't have a metabolism. There's no, where's all your building blocks? You're taking my building blocks. I had a plan for those. But now this is what you're doing. It costs energy to do all of this, to make more copies of the nucleic acid and the capsid and the spikes on the surface. All of this takes energy. It's all coming from this cell. So, you know, the headline will say viruses, they're all obligate parasites. Why? Because they can't do it on their own. They don't have any of the machinery. So no ribosomes. They lack most enzymes. They basically are hijacking a host metabolism. Okay, TSM, right?
That's okay. I figured you needed to get through.
Yeah.
So if it's a DNA-based virus.
Oh, we'll talk about that.
Okay.
Because it is different. It is
You know where I'm
different.
going.
No, it is. It's
Okay.
different.
Okay.
It's different. They're two different
I'll be
things.
patient. Thank
Okay,
you.
thank you. I mean, it's hard because I'm like building, you know. And the thing is, especially because you're familiar with the system. We've all
Yeah.
had a viral infection, so, but I just like, I can't imagine a situation where you have it. Okay. But they all have a host specificity. And this is called tissue tropism, and I'm sorry that's not written on here. tissue tropism. And so what this means is that they have a certain host range. So plants have viral infections, and they're, those viruses, they're after plants. We have some that are after bacteria. Bacteriophage, we talked about that, and we'll again, is after bacteria. And then there's those that are after animals. Unfortunately, sometimes the animal viruses, they're just looking for vertebrates. So this is why you can get avian flu, okay, or swine flu. It's because you share a lot of things in common. So the specificity of which cell is after, this tissue tropism, has everything to do between the fit on the spike, the viral protein spike, and the host cell receptor. Okay, that's really specific, and it's a 3D specificity. Okay. And so if we look at just 3 viruses as an example, influenza, it has on its surface H and N spikes. You've heard of the H1N1 that happened a while back. Right? They just get numbered every once in a while. I don't know what one is that we're concerned about. The H spike is the hemagglutinin spike. It's after the hemagglutinin receptor on the surface of respiratory epithelia, and that's how it enters. And it actually needs the N spike to pop out on the other side. So, if you've heard of Tamiflu, it stops the N spike formation, so it can't pop out. So that part's good. You can't stop it from getting in, but it doesn't release any extra. So it keeps your infection level low. Herpes simplex is after specific cell surface receptors that are only found on the oral cavity and in genitalia. And it used to be, back in the day when I took this class as an undergrad, that herpes simplex 1 was oral and herpes simplex 2 was genitalia, but things have gotten blurred in the ensuing X number of years, I won't not say. So now they just throw in the top, because they have found 2 in the mouth and 1 in the genitalia. So just like, okay, 1 of the 2. That's all it's after. HIV/AIDS is even more specific. It is only after a T helper cell. So that is a very specific cell of the immune system. You can be exposed to every single one of these by, let's say you dumped your hand into a vat full of either any of these. You cannot get the flu in your hand. Your hand will not start coughing. Right? Oh, that's not going to happen. Why? You don't have the receptors. The receptors do not exist on the epithelia of your hand, okay? Nor can you get herpes on your hand, nor can you get HIV on your hand, okay? What if you had a cut in your hand? And now the HIV gets in and finds the T cell in the blood, then yes indeed. Okay? What if you got herpes on your hand and then wiped your hand over your mouth? Then yes indeed. You got the influenza virus on your hands because you were using and opened the door or used the common keyboard at the library and then rubbed your nose, then yes indeed, you just put it exactly where it wants to go. Okay? So I just want us to recognize that they're really specific about which cell they're after. Okay? And it's based on that spike. The spike binds to a specific receptor. The mode of infection, okay, and I'm going to write this up here. The mode of infection is how it gets into the cell, okay? And it is not the same thing as transmission. Okay, students mix this up a lot. So I will have a page that says, the following are the general characteristics of virus: genetic material and a protein code. Then you're going to say it's DNA or RNA, never both. The protein code is a capsid. It can take on any of the following shapes. Okay, it's an obligate intracellular parasite. What does that mean? Well, it has no ability to make copies of itself. It has no ATP. It has no ribosomes. Okay, no metabolism for building blocks. Nothing. Right? Okay. It has a specific host range. What's that about? That's the spike landing on a specific receptor, tissue tropism. Okay? And now this, mode of infection. How does it get inside the cell? Because outside of the cell, it can do nothing. We just said that. They're obligate intracellular parasites. So this question is, how does it get into the cell? The problem is 30% of you are not really listening to me, and then what you'll say is, from coughing or sneezing or wiping your hands across your mouth. I didn't know. No, that's transmission. That's a different word. No points. No points. It's usually because there's 5 bullet points or 2 each. That's a 0. I got nothing for you. That's transmission. It's something else. Right? So, take heed. I taught this class a really long time, and I see all the places people mix up and mess up. Okay, so that's one of them. Don't mess up that. Mess up some more. All right. So how are you going to get in? Okay, so the mechanisms are going to be phage, you're going to land and inject. Inject. Okay. Now, receptor-mediated endocytosis, this is going to be what we're going to see for naked virus are going to enter through receptor-mediated endocytosis. And if you're thinking, I don't remember what that is, then you need to go back to the eukaryotic PowerPoint. Okay? There's like 3 slides about it. That's it. Okay, there's phagocytosis, there's penocytosis, and receptor-mediated endocytosis. Remember that receptor-mediated is really specific. You have to bind to this receptor. That's it. Not everybody gets in, just the one that lands on the receptor, yes? Okay, phagocytosis is different. These other 2 we can forget about, but I want us to know this. Fusion, right, will happen for the engulfed. What does it mean to be engulfed? What's on the outside of it? A phospholipid bilayer, right? Phospholipid bilayer. And your cells, what is on the outside of it? A phospholipid bilayer. And what happens when 2 lipids find each other in aqueous environment?
They repel.
No, they join
They
together.
join together.
They're like, hi, Fred.
You know what it is? It's actually water. I eat fruit. You guys go there. If you haven't tried this yet, please go home and do it. The dish of water, you just have to sacrifice 2 drops of olive oil. 1 over here, 1 over there. Just watch. It fuses into 1. Okay? That's fusion. So if it fuses like that, now it just dumps its contents into the cell. That is a way to get in. It needs to get into the cell. That is the main thing. That is not about wiping your hands. Okay? That's different. It's got to get in. So your question, what it can't tell the difference between a live or dead cell? Okay, it can't. If this cell is dead and not completely depleted yet, and you're a enveloped virus, then you're going to dump yourself into a system that can't replicate. It can't do anything for you. Okay? If you're naked and you land on the correct receptor, and the fusion requires a receptor as well. That cell can't do the process of bringing. Okay, so it's a dead end for that in either case. Yes, Amelia?
What causes fusion in like lipids? Because aren't they
Water.
both non-polar?
Yes, water does it. Water likes to play hydrogen bonding games, right? It can't do that if liquids are around because liquids won't play with them. So they're like, there.
Get out of our sandbox.
Yes, please,
So is
please
it-
do this. I don't have oil in the lab for you to play with.
It's
Okay?
a polar environment.
Yeah, I mean, I have oil for like the microscopes, but we're not allowed to play with them. Okay, Palmer first.
Is the envelope changing? Like, do naked DNA or naked virus get an envelope once they get in, and then also do enveloped virus then like change their envelope?
Well,
By
the
incorporating-
enveloped virus will change their envelope. Yes, they'll have the same spike, but I mean, what about the lipids? Aren't the lipids slightly different? You know, if they're like cardiolipin on there, you know, and it's totally depends. Which cell did you just jump in? Okay, we're not uniform, right? So, for the purpose of discussion, we're sort of talking about every eukaryotic cell as though it's roughly the same, but it's not, you know it's not. I mean, the lipids present on neurons are very different from the lipids present on hepatocytes, right, this kind of thing. So yeah, so the envelope looks different. The spike will be the same. And your question?
I was just gonna ask if you go over which one specifically can do each one, like I know the
Receptor mediated is gonna be for the naked, and then we'll just put an X on this, because I don't know why I just didn't say fusion, $1 billion. Okay?
Godzilla.
Alright.
Wait, what did you say that very last part?
Fusion?
Is facilitated diffusion?
No, I want you to put an X on these two.
Okay.
You know, let's do this. I'm gonna download this, and I will just fix it and then upload.
What about the phage? Is
No, that's correct.
that's, is that for both?
That's for bacteriophage, that's what they did.
Okay,
Okay, this is receptor-mediated endocytosis. I showed this picture before, I believe, when I showed receptor-mediated endocytosis. I showed you totavirus, and I said, do you think that that's what the purpose of the receptor was on the surface of the cell? Absolutely not. Has a completely different purpose, but it's taking advantage. Okay, it's taking advantage of that. All right, and then lastly, viruses self-assemble. So though they have to take over the host cell machinery and all of their ATP, and not all, but you know, a good fraction of the ATP, and they have to take over small building blocks for their capsid and for their nucleic acids, in ways that are not understood, the capsid and nucleic acid come together. It's not understood, but it has been seen in the lab, and I could not download the picture, so I drew it myself, you can see. So this is the red is supposed to be the nucleic acid, and these hexaderons are the capsid. And if you put them in a test tube and come back later, the nucleic acid's inside the capsid in a way that, like, it's independent of the cell. There's no cell involved. This is a cell-free system. It's just a test tube. So this is not understood. So they do this part on their own. The cell is not equipped for this procedure, but they can do it on their own. Okay, self-assembly, yes.
How long does that process take?
Not very long. Not very long. Okay, so what you're seeing here, and again, it's a nanometer range, this is HIV. And so the naked ones are slightly different. They're gonna burst out of the cell, but the enveloped ones bud. You see how it's like sort of budding off like that? Okay. Unfortunately, see, this is the budding that you're seeing here, and here's the budding that you're seeing here. Binary fission, we start with one cell and end up with how many? Two. With this, one infected cell will release thousands. So, I know we described bacterial growth and the growth curve, that steep thing, we called it exponential, and it most certainly is. 2, 4, 8, 16, 32, 64, and I think it's big fast, okay, yeah. Except that with virus, 1 becomes 1,000. 1,000 become 1,000,000. It's like they can't even be put on the same scale. Do you understand that? Okay, so this is a big deal. That's why the Tamiflu, stopping the release, is a big deal. Okay, you're not stopping 2 things from getting out, you're stopping thousands from getting out and doing the exact same thing. Yes?
Self-assembly is something that they're researching as a target for keeping viruses from replicating.
There's a lot of work on virus in all different directions. Yes. Okay, so those were like the general characteristics of virus, but each virus is going to be distinct in its nucleic acid. Is it DNA? Is it RNA? If it is DNA, what is it? Is it a single strand? Is it a double strand? Is it a circular strand? If it's RNA, is it a single strand? Is it a double strand? Is it carrying with it associated proteins? Okay, capsid structure. Okay, are you helical? Are you icosahedral? Are you bullet shaped? Are you complex? Host range. Which cell do you like? Okay, you're after respiratory epithelia, you're after T cells, you're after genital or oral cavity. Okay, there's all kinds of viral stuff. There's hepatitis that's after hepatocytes, right, liver. So you can't be really specific about that. And then mode of infection, and what does that mean again? How it gets into the cell. So it's either getting injected for the phage, but if it's a naked, how does it get in? Receptor mediated endocytosis, and then if it's enveloped, it's fusion. In either case, we require the spike to land, okay, on the correct cell. And I ask these questions not because I'm expecting that literally 4.5 minutes later you memorize what was on the previous slides, but because I know that many of you are going to re-listen to this lecture. And here comes the question, and this time you got it. Okay? It's all like mind games, because then you're like, yeah, you know, right? Like, that's a good thing. Okay? And if you like still can't answer the question, then you just go back and look at the slides and come back and answer the question now, right? Like, it's all good. All right. So here's our general information. So you should be able to flesh this page out. I know I'm pretty sure I have it on the worksheet. And so here's slide 31, and you want to be able to do that. Okay? So flesh this out. All right. So if you're a virologist, then they say that a virus is a different form of life. But if you're not a virologist, then you say that viruses are non-living. And in either case, we have to really pause for just a moment and recognize that these are human constructs. The world does as it will, and we chase behind it and try to shove everything into a box, which is problematic on all fronts, quite frankly. Okay, because you will invariably come up against something that refuses categorization. And what are you supposed to do? What am I supposed to do? So I will have this question. Why do you think they're alive? Why do you think they're not? But I want like 2 pieces of evidence or thoughts behind it. Okay? So right now I'll lay out the non-living section. You can certainly take something off of this, but something else might occur to you too. Okay? Well, we'll start here, cell theory. Cell theory, which has been around for a very long time, says the cell is the smallest unit of life. All living things carry out 8 different functions, or have 8 different characteristics, maybe is a better way to say it, right? That they are all cellular, right? That they all have homeostasis, that they all grow, develop, and reproduce, right? On their own, okay? But what about, and have a metabolism? There's no metabolism here, all right? But then, what about the evolve? Is that a problem for us? Do vaccines that are made right now against the flu, do they work next year? Maybe. It changes, doesn't it? And when it changes, that's evolve. Well, shoot, it evolves. Okay, well, okay, so the problem is we have really bad language. Living, dead, and non-living. This is non-living. Come back next year, it's gonna look the same. Okay, because there's no ability to evolve in any way from the stapler. Virus, that's our issue. Okay, COVID-19, remember we all lived through it and was like, oh, the Lambda variant. It's like, okay. Rich. So they're acellular. They are not cells. To be a cell, I gotta find within you 4 structures: plasma membrane, ribosome, cytoplasm, and double-stranded DNA. Okay? They might come in with a double-stranded DNA, maybe. Only some of them. The rest of it, no, and no. Okay? Can't replicate outside of a host. Oh, lack all cellular machinery. Sorry, I didn't speak that. Okay, so they don't have ribosomes. They don't have most of the enzymes. They have no way of making ATP, right? They have no way of breaking down materials. They have no way to do that. Back to my example with the 2 slabs of meat, you squirt Bacteria on a dead slab of meat, it's like, yeah, I can break all of it down, all of it. Virus, it's like, who's gonna do this for me? They can't do it, right? Okay. Can't replicate outside of the host. So what does that mean? Well, okay, so we spilled some virus on this table. Ooh, oh my goodness, the virus. You know, but there's proteins and lipids on the table too. So, right, like, it just, okay, so it'll be there for a little bit. I mean, we're just looking at rates of decay now. I mean, it's not doing anything. I'm not that afraid, okay? Bacteria pose a completely different problem, okay? And I will say this, there are definitely viral infections that are gonna kill somebody, absolutely. But in terms of the virus and the bacteria, to the virus, if it kills you, it just lost. Okay, that's as far as it's gonna get. Ebola, as bad as it is, you know, you don't feel so good, and then bleeding out of every orifice the next day, that first off keeps everybody away from you, and secondly, like, it's over. It's over. That cannot propagate. To bacteria, the sooner you and your pesky immune system are out of the way, the better. Okay, because you're just a big bag of 4 carbon compounds as far as they're concerned. Okay? Am I skipping somebody's question? Okay. Do not always have their genetic material in the form of double-stranded DNA. All living systems, this is how they do it. Okay? And they don't have all the characteristics of living things. So that's something that we have to recognize. That I think they might be some other form of life. You know, I'll let you work on that. I'll let you work on some of these thoughts. Yes?
So a virus by itself with no living host will decay slowly. How long does that decay process take?
Well, so whatever number you would get on something, you can probably bet there's a 12-hour gap where they're just like CYA. Okay, so for HIV, they'll say 24 hours. Which probably means within 12 it's gone. Okay? But,
How about
you know, put it way out there, right? So, I mean, this is how we do things. So, I mean, it's just the way everything is, right?
Following on that is there's been we've found that like viruses have been frozen in ice.
Yeah, that's different.
How is that different?
Well,
Because it won't.
so, okay, I can arrest everything from happening, not stop forever, but slow it down if I put it on ice, right? I can stop bacterial growth by putting it on ice, yes? Okay, the same is true for decay.
You just slow the decay, but it's not necessarily stopping the decay.
Correct.
I'm just curious if the
Are you curious if the warming of the globe and the permafrost?
I would love to
Yeah,
see
well, something
like
killed
that
those animals.
Yeah.
You've never seen that something.
Well, we're gonna
So,
see it
yeah.
soon.
Yeah, I'll be long gone, yes.
Things to look forward to.
Hey, Dan, as far as on this topic, are we deciding?
Yes,
Okay.
yeah, it's kind of open-ended, I like open-ended stuff,
Okay.
okay, sort of like, what do you know? Yeah, okay. And I'm often surprised by the students' answers, okay, just like, okay, oh, okay, hang on, I'll say that later, I want to go back here. Okay, so now that brings us to virus part 2, we do have some time.